hands, who has the policy acknowledgement done? 47 out of 47. That's what I like to see. It's almost everybody. Fantastic. If you haven't done it yet, make sure you do it as soon as you can. You don't want to forget. As your kind of weeks go on, we get into the semester, you get busy. I don't want you to forget to do it. So please do it as soon as you can if you haven't done it yet. Is the sound like echoing today? Is it different? Or is it okay? It's fine? All right, it's weird for me. That's fine. No problem then. So today's topic is really fun. This is one of my favorite topics. I'm going to say that like at least 6 times this semester. But this is the first time I'm going to say it. This is one of my favorite topics. And the reason for that is that this topic is like ongoing right now. Like, there's not a lot where you don't even get into like how photosynthesis works. There's not a lot of cutting edge research in terms of that. This is going on right now, so this is a little bit different every year, which is super fun. But we still have to finish up biological molecules, so we're going to come back to this in just a few minutes. We don't have a lot left to cover, but we have to talk about proteins. So we're going to do that. We're just going to do that last bit from the previous topic. And then, I say this every time because I'm an optimist. I don't know if we're actually going to do it, but I think there's a good chance we get caught up today. And if not today, then next time, because the next lecture topic is a little bit shorter as a standalone, so if there's any spillover, we should be fine for next time. So we're just going to keep going through it. We'll see how far we get. So let's pick up where we left off last time with proteins. So this was in biological macromolecules, section 2 from last time. We had done macromolecules and carbohydrates, and we finished up with lipids. So now we're going to talk about proteins, and then we're going to mention nucleic acids, then we'll get into today's actual topic. All right, so proteins are polymers. Just remember, all of our macromolecules are polymers except for lipids. So proteins are polymers, and the monomers are these things called amino acids, which are abbreviated AAs. Anytime you see AA up here, that means amino acid. And all amino acids have the same basic structure. There's a bunch of them. There's a whole lot of different kinds. But they all have the same basic structure. This is the structure of amino acid. You need to be able to recognize this structure. You don't need to know any specific amino acid, but you need to recognize the general structure of amino acids. They all have the same parts. Okay, so all amino acids have a central carbon called the alpha carbon. That's this one right here. And because that's a carbon, there are going to be 4 things attached to that carbon. Every amino acid has 4 things attached to it, and for every amino acid, 3 of those things are identical. So we have, of course, a hydrogen atom right here, just a boring old hydrogen atom just sitting there doing hydrogen-atom things, which is not much of anything. We have on one side an amino group. So think back to our functional groups from last time. You should recognize the amino group, NH2, and remember they'll ionize to NH3, in cell. Then on the other side, it's always drawn like this, by the way, with the amino group on one side, and on the opposite side, we have the carboxyl group over there. So remember, that's acidic. It's going to lose that proton, and it will be negatively charged in biological systems. And the last thing is the only thing that differs between the different amino acids is this R group up here. That's also called the side chain, and that is going to be different across the 20 amino acids that we use to make our proteins. So cellular life, a lot of variance on this structure. There's all big, small, all different properties. I'm going to show you some examples in a minute, just to show you how they can differ in their properties. The key thing I want you to see before I show you that is that all of the amino acids are the same, except for that one part. So it's the differences in that side chain that are going to dictate the properties of the amino acid. And that's important because, again, be thinking about emergent properties. When you put those amino acids together in a big long chain, that chain is going to have different properties depending on the types of side groups that are present and the locations of those side groups within that big long chain of amino acids. So you're going to get emergent properties of the large molecule that can't be found within the monomers, but are based on or due to the composition of those monomers. Everybody okay with that? The side chains do all the work here in terms of dictating the specific properties. So let's look at what these can be. Now, don't memorize this, don't write this down. This is to show you the variation in structure. Some are very small, some are very large. They have all different properties. They're color-coded according to their properties. So the yellow ones up here, those are non-polar. Those side chains are hydrocarbons. So they don't have any polarity, they don't form hydrogen bonds or anything, so they're going to be hydrophobic. You can have polar side chains like hydroxyl groups or sulfhydryl groups in here, so those are going to form hydrogen bonds. And then down here, in pink and in blue, you can have acidic or basic amino acids. So not only will they be polar, they'll actually acquire full units of charge. They'll be positive or negative, and then they can even engage in ionic bonds when they're charged. So within a protein, within a lot of amino acids put together, a big chain, these are going to interact with each other in somewhat predictable ways based on their different properties. The hydrophobic ones are going to stay with other hydrophobic ones. The polar and ionized ones can interact with each other either through things like hydrogen bonds or ionic bonds. So these variations have big implications for the structure of the protein. Now, you don't need to know, again, you don't need to know any specific amino acids here. If you take biochem, you will just, by doing biochemistry things, you will learn most, if not all, of the amino acid structures. You may have to, but just by doing stuff a lot, you're going to learn these if you take biochem. If you don't take biochem, you may not learn, and that's okay. You don't have to. We okay so far? Amino acids? Okay, so the way we build a polypeptide, a big chain of amino acids, is by connecting those amino acids with bonds called peptide bonds. And the peptide bonds occur between the carboxyl group of one amino acid and the amino group of the next amino acid in line. So that chain has all the amino acids kind of oriented the same way, all the amino groups on one side of them and the carboxyl groups on the opposite side. And the way we make a peptide bond is a dehydration reaction, just like always. Anytime we're doing polymerization, we're doing the dehydration reaction. So you can see right here, you've got the OH part of a carboxyl group, and you have one of the hydrogens of the amino group. Those are going to react together to form H2O. And this bond's in blue right there between the carbon of the carboxyl group on one amino acid and the nitrogen of the amino group of the next amino acid. That bond right there, that's your peptide bond. So remember last time, anybody remember what's the bonds that we find in carbohydrates that holds the sugars together in a polysaccharide? Anyone remember that one? Just shout it out if you got it.
Glycosidic.
Glycosidic linkage, good. What was the one that we saw in lipids that connects the glycerols and fatty acids? Remember that one? That was an ester linkage. Here we have a peptide bond. So each type of macromolecule has a characteristic type of bond between its components. You should know peptide bond, proteins. You should know that. Okay? So that is a peptide bond and what it does and how it works. It's all about that dehydration reaction. So we take a bunch of our individual amino acids, we attach them to each other using peptide bonds. We get a big, long chain. What do you think that big, long chain is called? We have peptide bonds, and we have a lot of them. What's a word that might mean a lot of peptides? Polypeptide. Many peptides, right? So polypeptide. It's a big, long chain of amino acids. Hundreds, thousands, tens of thousands in a linear sequence. That's it. That's all peptide is. Just a big, long chain of amino acids. That's it. It's not more complicated than that. One little bit of a distinction we need to make here is that a polypeptide is not yet a protein. A polypeptide, that term just refers to a long chain of amino acids. So it's not a protein yet if you just have a long chain of amino acids. You have to fold it into its correct functional shape. At that point, that molecule can be called a protein. But when it's just a big long chain, the term for it is polypeptide. Now, oftentimes you'll hear people use the two terms interchangeably when referring to actual proteins. That's fine. It's not super precise, but it's fine. But you can't call just a chain of amino acids a protein. It's not a protein until it gets folded up. So just be aware of that distinction. A protein is a more specific term than polypeptide. So the way we get a polypeptide to become a protein is we have to go through several layers of folding to get to its final structure. Fortunately, the names for these different levels of protein structure are very intuitive and easy to remember. Okay, so the primary structure of a protein just refers to the polypeptide. It's just the linear sequence of amino acids. That is the primary structure. Sequence of amino acids, primary structure. The secondary structure refers to local small-scale folding between nearby amino acids. So if you have a big long chain of 1,000 amino acids, maybe you've got amino acid number 5, and then the chain kind of loops around, and amino acid number 10 forms an H bond with amino acid number 5. That local folding, that results in your secondary structure. And there's a few different flavors of secondary structure. You don't need to know the specifics. There are these things called alpha helices, where it kind of wraps around like a spiral staircase. There are beta pleated sheets, where it kind of zigzags back and forth, and the chains adjacent to each other as they wind back and forth form H bonds. You don't need to know the specifics of those. Just know that secondary structure refers to localized folding between amino acids that are relatively near to each other within that polypeptide chain. Yeah. Primary structure is just the polypeptide. It's the linear sequence of amino acids. So if you just lay out 1,000 amino acids in a row with no additional folding, that polypeptide has primary structure and nothing else. And then you do little localized regions of folding, that's going to be your secondary structure. You take that whole big long chain, fold it up, and wrap it around in a complex 3D shape on itself, that's your tertiary structure. This is large-scale folding that spans long distances in that polypeptide chain. So like, for secondary structure, the example I gave you is an amino acid number 5 was there, and then it kind of loops around, and amino acid number 10 forms an H bond with number 5. That's going to be secondary structure. But you can also have that chain go off and do other things, and then it loops back around, and amino acid number 900 forms an H bond with number 5. That long distance, large scale folding, that's going to be your tertiary structure. Yeah?
So does each structure have a different then?
Yeah, we're going to get there, don't worry. Multiple amino acids is polypeptide. A polypeptide folded in its functional shape is a protein. You have to do the folding to become a protein. Okay. Now, before we move on to our last level here, I want to just draw some distinctions between secondary and tertiary. There are a few things here I want to point out. One, I've already pointed out, is the distance. Right? It's local versus long distance. Okay. In fact, why don't I write the table for this? Hypothetical. Right? Local versus long distance folding. The second thing, the second distinction between the secondary and tertiary structures is the part of the amino acid that's involved. The secondary structure involves what we think of as the backbone of the molecule. So it's a lot of the amino groups and the carboxyl groups that make up the actual backbone of that chain interacting with each other. So it's short distances, it's the backbone, whereas the tertiary structure, it's long distances, and it's the side chains, those R groups, that are interacting with each other across those long distances. So it's different distances or scales, different parts of the amino acid, and the last one is the different types of interactions. Secondary structure is basically entirely hydrogen bonds. So you have like your carboxyl group and your amino group, you still have the C double bond O when there's a peptide bond, and you have the N with a single hydrogen. Both of those things, that oxygen and that hydrogen, are polar. They are partially charged. So they can form H bonds with each other, but that's it. They don't do other types of interactions. It's just H bonds. Whereas here, because of all of the different types of side chains that we have, polar versus non-polar, acidic, basic, you can have hydrogen bonds during your tertiary structure. You can also have ionic bonds. You can have covalent bonds. You can have like van der Waals, like London dispersion forces. You can have H bonds. You can have anything. Any type of interaction or bond that we've talked about can play a role in tertiary structure. So we have three distinctions between secondary and tertiary. The distance, short distance versus long, the part of the polypeptide that's involved, the backbone versus the side chains, and the types of interactions involved, hydrogen bonds versus any type of interaction that we talked about. Were you okay with up to tertiary structure? Is everybody okay there? Tertiary structure that gives a protein its like functional shape, where it's going to have like a groove, where it's going to grab onto something and cut it, or whatever. But some proteins don't stop there. Some proteins are done right here. It's a single polypeptide, it folds up, it's good to go. Some proteins involve more than one polypeptide chain. Example, this is hemoglobin, which is 4, the protein that carries oxygen in your blood. That has 4 polypeptide chains. So that protein has quaternary structure. That's where you have more than one polypeptide that assemble together into a final complex. And you don't have the final functional protein until you have all of the component polypeptides present. Again, not all proteins have that, many do. A lot of big complex proteins in our bodies and our cells use quaternary structure. It's more than one chain. Yeah?
What's the most common type?
All proteins do primary, secondary, and tertiary. Some do quaternary, I'm not sure of the fraction. But all proteins at the very least go up to tertiary. If it doesn't have tertiary, it doesn't work. Because structure dictates function. If it doesn't have the right shape, it can't do its job. And when proteins lose, question, go ahead.
Sorry, could you explain quaternary one more time?
It's multiple quaternary is just more than one polypeptide. That's enough of a distinction from the others. We don't need to worry about anything else. Now, this is all about the proteins getting and maintaining their shape. When proteins lose their shape, they lose their functionality. There's a specific word for that, and it's called denaturing. When a protein unfolds, when it loses its tertiary and sometimes secondary structure, depending on the situation, we say that that protein has become denatured. When you lose the structure, you lose the function. Now, an important thing about this happening, when a protein denatures, a lot of times you don't break a single covalent bond. It's all just hydrogen bonds and ionic bonds that make your secondary and tertiary structures. But you're not breaking the polypeptide backbone, you're just kind of unwinding that three-dimensional shape. So a lot of times a protein can denature, and then it can be refolded back into its correct shape. So once you don't break any of the peptide bonds, you just kind of fold it back up. We're going to talk more about protein structure and how it matters for protein function in like a week and a half when we do metabolism, because we're talking about enzymes and active sites and all that fun stuff. So we're going to cover this in more detail in just a little bit. We'll get into kind of the specifics of why the structure matters so much and what we can accomplish with our proteins. This is something we'll talk about when we get there, just why would a protein denature, what can cause that. Anything that's going to disrupt interactions between charged regions, so changes in pH, high salt content, high temperature, anything like that can cause a protein to denature. And we're going to see, again, we're going to see effects of that in specific examples when we get there. So this is just kind of an FYI. We'll get there in a couple of weeks. Last thing we're going to do with proteins is just functions. So anyone want to shout out some protein functions? What proteins do in your body? What do you think? I heard the word enzymes very quietly. Somebody said it though. Nice. Yeah, enzymes. They speed up chemical reactions. Yeah. Any other ideas? Structure. Yeah, we got a lot of structural proteins, right? A lot of structural proteins like connective tissue and like layers of fibers in your cells and your like on your skin, for example. Yeah, absolutely. Basically, proteins do everything except heredity. That's it. That's what they do. It's everything except heredity. So they're involved in structure. They're involved in signaling. They are enzymes, of course, to speed up reactions. We'll see countless examples of that. They're involved in defense. Way at the end of next semester, we're going to do the immune system. We're going to see some really cool defensive mechanisms that we have. They're involved in transport, either across a membrane or in the bloodstream or in some other way. A lot of examples of transport proteins. So the only thing that proteins don't do, basically, is heredity, transmitting genetic information. That's other stuff does that. Other than that, it's all proteins. So that is it for proteins. We're caught up for proteins. What questions do you have with proteins before we change gears? We're going to get to this the next slide, don't worry. We're going to get there. So else can you, oh yeah, over here. Yeah, go ahead.
So when you form the peptide bond and you do dehydration synthesis, how exactly does that happen?
Don't worry about how the peptide bond is formed. We're going to do gene expression later in the semester. We'll talk about how ribosomes work and how that happens. Don't worry about that. Yeah, you should just know that it's an example of dehydration synthesis. The resulting bond is called a peptide bond, and you should be able to recognize a peptide bond. If I show you a picture of 2 amino acids, you should go, aha, that's 2 amino acids with peptide bond. Secondary structure is local backbone H bonds. Tertiary structure is long distance side chains, every interaction we talked about. H bonds, ionic, covalent, everything. Oh, one more. Yeah, go ahead.
When a protein like denatures, will it restore back to its normal shape by itself, or put it on condition?
When a protein denatures, will it spring back to its normal shape by itself if the conditions go back? Sometimes yes, sometimes no. We do have proteins in our body called chaperones that help other proteins fold up into the correct shape to kind of help them along if they can't do it themselves. All right, this is all we need for nucleic acids for right now. Nucleic acids, DNA and RNA, we're going to talk about it later. Our whole second unit is on like genetics and heredity and all that fun stuff. So don't worry about it. Just nucleic acids, DNA, RNA, coming soon. You don't need to know anything else about it. We'll get there. You good with DNA and RNA? That's all we need. Literally 4 words. Nucleic acids, DNA, RNA. Done. Ta-da! The easiest biological molecule. Around. Okay. We ready for origins of life? This is a super fun topic. Let's do it. All right, so now, new Bibles form, new page in your notes, new topic. How are we doing on time? We're doing fine. So we have 2 topics today. Our first topic is the process of abiogenesis, basically what we think about how life formed. We have 3 outcomes for that section. Compare and contrast the requirements, like on an early Earth, for life to form. Compare and contrast 2 hypotheses. Yeah, 2 hypotheses. There's more than that now, but we're only going to talk about 2 hypotheses for one step in this process called abiotic synthesis, and then we want to sequence the overall process of abiogenesis. And as we'll see, it's going to have 4 steps. So we're going to sequence those steps. We'll go through each one. Once we get to, okay, cellular life is here. We accomplished that. Now we'll talk about the brief history of life on Earth. I want you to sequence the early history of life, starting about 4 billion years ago and taking us up to approximately 500 million years ago. Is about how far we're going to get. And there's only a few major events that happen there. And I will tell you right now, when we get to this section, I don't want you to like memorize the specific years that things happen. Oh, this was 1.8 billion years ago. This was 1.5 billion years ago. I don't want you to memorize that. What I want you to know is the order in which things occurred, because they have to happen in a certain order, right? You have to have oxygen in the atmosphere. At some point that happens. You need to have photosynthesis before that. There's something else that develops later that you can't have without oxygen in the atmosphere. So the sequence is what matters. I don't care if you know the years. The years don't matter. It's good to have an idea. But it's the sequence that matters and why it has to be in that specific sequence. So there's lots of hypotheses when it comes to the origins of life. There's, we're only going to talk about this one specific idea. But there's an idea we're not going to spend time on called panspermia. Has anyone heard of this idea, panspermia? This is the idea that life on Earth came from a different planet. And there's like 2 flavors of panspermia. There's like the kind of defensible one, which is a fringe idea, but like sort of reasonable. That like, well, Mars probably had life at some point. And if like an asteroid hit Mars and ejected stuff from Mars, then maybe some of that stuff landed on Earth, and that's how cellular life got on Earth. Like, that's the kind of more reasonable version of panspermia. The unreasonable version, aliens. Like, did not like put life on Earth on purpose because we're just like a petri dish, and they're watching us evolve, right? That's the kind of out there version. Okay, we're not gonna worry about this. We're not gonna worry about this. By the way, this guy apparently has like, at some point someone pointed to me, I've since lost it, but somebody like pointed me to his Twitter feed, and he's like, he doesn't believe any of this. Or like apparently he just like kind of like getting old, I don't know. But alright. So the idea we're gonna focus on is the title of this topic, abiogenesis. We're gonna break that word down in just a minute. This had to have occurred more than 3.7 billion years ago. Now, Earth is about 4.5 billion years old, give or take a few hundred million years. And we know that the cellular life existed by 3.7 billion years ago because of formations like this. This thing is called a stromatolite. You don't need to know that word, you don't need to write it down. This is FYI. This thing is called a stromatolite. And what it is, it's basically layers of fossilized microbes. So they grow, and subsequent kind of generations grow on top of them, and they compress and they fossilize over time. It's basically like a microscopic coral reef. You kind of think about it like that. And we know exactly what this thing is because we can test its chemical composition, and we can find stromatolites in the real world. If you go to Australia, and you can like swim in the water, and there are stromatolites right there. Don't touch them, don't pick them up, you'll get arrested. You can't do that. But if you like have a scientific permit and they allow you to do it, you can go and find a modern stromatolite and cut it open, and you can compare the kind of just what it looks like and the chemical composition to the 3.7 billion year old version, and they're like the same. So we know that these things are formed by populations of microbes 3.7 billion years ago. So how did those cells get there? How do we go from a planet that's like all volcanoes and asteroid impacts to cells within a few hundred million years? That's what we want to consider. And this process is called abiogenesis. We can break that word down. A, the prefix A, means without. Bio means life. And then genesis is origin. So the without life origins for living things. How do you go from a planet without cellular life to a planet with cellular life? That is the process of abiogenesis. And we are going to have 4 steps in this process that we're going to go through. Okay? And now, if you were to like look online for information about this, you are going to find so much more than what we talk about in this class. So I want to be very clear. What we talk about here, this is what I want you to know. There's other versions of this, there's other hypotheses, there's like a lot of research going on right now into this stuff, which is why it's so much fun. This is the sequence I want you to know. Is everyone clear on that? This is one of those like cutting edge topics. So you look around on your own time, you're gonna find other stuff. Okay, so our starting point is our abiotic synthesis of monomers. We can get the organic molecules. Then we have to build organic macromolecules. We have to make the things that we just talked about, things like proteins and lipids. I see you, don't worry, I'll get you in a second. Once we have those macromolecules, we're going to form structures called protocells. You're not going to know what those are yet, and that's okay, we're going to talk about it. And once we have protocells, we have to have self-replication. Those systems have to be able to replicate themselves. And at that point, you're really close to having cellular life. There's an important component that you'll see is missing by the time we get there, but we're going to be pretty darn close. Now, I want to say up front that in this process, these are the steps in this sequence I want you to know. In real life, if this is how it happened, steps 3 and 4 probably happened around the same time. But I want you to know it. You're 1, 2, 3, 4 in that sequence. So we're clear on that? This is what we're doing. Question? The word again, abiogenesis. Yeah, without is the A is without, bio is life, and genesis is origin. So the without life origin for cellular life, abiogenesis. So what we're going to do most of today is going through these steps. This is one of our big outcomes for today. Sequence the process of abiogenesis. This is it. This is your flow chart for sequence the process of abiogenesis. In here, in part one here, we are going to talk about both the requirements for this process, and we're going to talk about two of the competing hypotheses for this first step. And then 2, 3, and 4 are actually pretty quick. Yeah. Sorry, say that again a little bit louder. Don't worry, we're going to define all these terms. I wouldn't expect you to know all these terms yet. We're going to get there, don't worry. So let's start with abiotic synthesis, right? So our starting point is that living things had to come from an environment without living things. And again, that word is abiogenesis, without life origin. So the molecules that are going to be the basis for that, all of the things like lipids, amino acids, and whatever else, the molecules that are going to form the basis of these first cellular living things have to appear spontaneously in the environment. And the spontaneous appearance of those molecules is called abiotic synthesis. So again, A is without and biotic is living. So the non-living synthesis of molecules like amino acids and lipids. So that's our first step in the process. We have to go from an environment that doesn't have any, lipids, for example, to an environment that has lipids in it. And it has to happen abiotically because there's no cellular life that already exists to make those things. Right? So that's why that abiotic synthesis has to be our first step. Is everybody okay with that? See why that's the case? So we're going to start with the smallest molecules first and go from there. Now, you can't just do abiotic synthesis. There's a lot of requirements for that. A lot of hypotheses about how it would have happened. So we have 4 requirements. You have to meet all 4 of these requirements in order to achieve abiotic synthesis. First, you can't have any free atmospheric oxygen. The atmosphere has to be low in oxygen. Why? Because what do we know about oxygen, based on what we've talked about over the last few days? It has really strong what? Just shout it out. Really strong electronegativity, yeah. Which means that it's really reactive. So if there's oxygen present as other things are going to start to react and form into larger molecules, oxygen tends to come along and steal electrons. And that means breaking bonds. So if you have oxygen present in the atmosphere, you can't build bigger molecules spontaneously. They're all going to break down because they're going to react with the oxygen. So you need low atmospheric oxygen. We're going to see how we got all the oxygen we have. We're going to see that a little bit later today. But our starting point is low atmospheric oxygen. And we can look at rocks that are billions of years old, and we can look at the composition of things and be like, yeah, okay, there's clearly not oxygen present at the time. Second thing is you need an energy source. Because there's no cellular life present, so we're not making anything like ATP, for example, the kind of the energy currency of cells. So all of your energy has to come from the environment. Fortunately, the early Earth was a highly energetic place. Lots of volcanic activity, geothermal activity, hydrothermal activity, as we'll see. Plus you've got lightning, you've got UV radiation from the sun, because atmospheric composition is different, so more of the radiation reaches the ground. So you have a lot of different sources of energy that are going to put energy into the molecules that are present, which will enable them to carry out various chemical resources without needing some cellular energy source. Okay, so we need an energy source to do the chemical reactions, and the environment was full of such chemical sources. Third, if we're going to build things like lipids, amino acids, nucleic acids, we need the raw chemical components. So you need a source of carbon, oxygen, and nitrogen, and hydrogen. Now obviously O2 gas is not going to work, but we probably have a lot of CO2, plenty of water, carbon monoxide, hydrogen gas, nitrogen gas, right? You need the raw materials that are going to contain those big 4 elements. And you also probably need some sulfur in there. There's going to have to be some phosphorus in there. You need just the basic chemical building blocks. And on top of that, there's definitely going to be some ions, like metal ions, things like nickel or iron. Because even today, metal ions are catalysts for various chemical reactions within our cells. So you probably needed some metal ions in various, doing various functions for those earliest chemical reactions. And of course, all of this is taking place in liquid water. You had to have liquid water in order for this to happen. This is why we're like pretty sure that there were living things on Mars at some point, because we know there was liquid water and a much denser atmosphere at some point in the history of Mars. Pretty good indicator that you've met the conditions for cellular life. The last thing we need is lots of time, because we're not doing these reactions in a lab. We're not purifying the ingredients and doing them in a flask, in a lab. We are doing these things in the environment where there's lots of spurious interactions and side reactions, and you take 100 steps forward and 99 steps backwards. And then you take 100 steps forward and 99 steps backwards. And that's what this process is going to look like. So there was this period in the early history of Earth, a little over 4 billion years ago, called the Late Heavy Bombardment, where we just got pounded by asteroids. And the consensus is that you couldn't have cellular life during the Late Heavy Bombardment. Think like killing the dinosaurs with that asteroid, but like a million of those in the span of a few tens of millions of years, right? Like just nothing. So we know we have like fossil evidence of early cellular life 3.7, 3.8 billion years ago. We got late heavy bombardment a little over 4 billion years ago. So you end up with this range of like 2 to 400 million years is kind of your sweet spot. That's your time frame where all this stuff would have happened. Now that in the span of billions of years maybe doesn't sound like a whole lot, but in any kind of absolute sense, 200 million years is a ton of time. So that that fulfills our requirement for we need lots of time for this stuff to happen, and there was that period where you had time for it. We okay with the requirements for abiotic synthesis? This is just what we need to start building the basic building blocks. Are we okay with that? All right. All right. So how do we do this? How do we build things like lipids or carbohydrates or whatever? Well, there are 2 general hypotheses for abiotic synthesis. There's actually more than that, depending on who you ask and how much time you have to listen to that. You could go down some rabbit holes with this, but we're going to talk about 2 of the main hypotheses. The thing I want you to get out of this is that these hypotheses are pretty similar in terms of what happened. They differ primarily in the where. And because they differ in the location, they also differ in terms of the energy source. But in terms of like the nuts and bolts of what happened, they're relatively similar to each other. If you ever say that to somebody who studies this stuff professionally, they will get mad at you. But for our purposes, they're relatively similar except for the location and the energy source. So the first of these hypotheses, called the Oberon-Haldane hypothesis, which is named for the 2 people who came up with it, A.I. Oberon and J.P.S. Haldane. They arrived at this idea more or less independently in the 1920s. And the idea here is that life first appeared on Earth in shallow water, so like near the surface, exposed to the atmosphere. And if you've ever heard the idea of like a prebiotic soup, who's heard that phrase before? It's not really a commonly used phrase all that much anymore, but if you've ever heard that phrase, it's referring to this idea that life formed in more or less shallow water near the surface rather than like deep oceans. It's the Oberon-Haldane hypothesis. Now this was tested pretty extensively and has been, and there's ongoing experiments in all of this. But the first really famous test of this hypothesis is the Miller-Urey experiment from 1953. Has anyone heard of this one? The Miller-Urey experiment is like one of those famous experiments. So they set up, it was Stanley Miller and Harold Urey, and they set up this apparatus basically to simulate an early Earth environment. So they basically had an ocean that they heated up like some like salt water basically. You get the water vapor in an atmosphere with carbon, nitrogen, and hydrogen. And then they used some electrodes to spark it. That was their energy source, like lightning. Right? That's the energy source they used. Then they condensed it out and collected their samples and saw what kind of molecules they could isolate in their samples. Pretty simple experiment. Right? Mix the ingredients you think together, zap them with some electricity, collect it out, see what you find. Turns out we found some really interesting stuff, like amino acids. A lot of amino acids. Found a bunch of amino acids, including some amino acids that we don't even use like in cells. We don't even use some of the ones that they found in proteins, but there are more than 20 amino acids. We just happen to use 20 of them. So they found a whole bunch of organic compounds, including amino acids, and this is basically like, okay, plausible idea, good enough. Now that was 70 years ago, and as you can imagine, we didn't just stop there. There's been a lot of work since then. Is everybody okay with Miller-Urey experiment before we keep going? Okay. Been a lot of work on this since then. So these experiments have been repeated under lots of different conditions. So there's some debate over whether the atmosphere would have been neutral or would have been what's called a reducing atmosphere. We'll introduce the concept of oxidation and reduction later when we get to metabolism, but basically just different atmospheric conditions. And we found a whole bunch of different things depending on the materials you start with and the conditions. So for example, you can find DNA or RNA nucleic acid bases, not the whole nucleic acids, but the actual base part, the ATCG in your DNA, just that part of the molecule. You get all the amino acids we use, various lipids and sugars, and if you have phosphorus, you can get ATP. And that's really nifty because what is ATP? We'll cover this in a few weeks, but what's ATP good for? That's energy in your cells, right? So if you start making ATP, now you don't need an environmental energy source anymore, because now you have a chemical energy source. It's a short-lived chemical energy source. It's not stable, you can't store it. But you still have it, you're making it, and that's really useful. All right, so some really interesting results there. They also actually went back decades later and reanalyzed the original samples from 1953, because way back in 1953, our kind of methods for detecting what was in that stuff, measuring like the amino acids and sugars and whatever else, was actually relatively primitive. We have much more sensitive ways of measuring that stuff now compared to back then, and it turns out they missed a bunch of stuff. So this is 1953 versus 2008, the number of amino acids. They originally documented like 7 or 8 of them. They subsequently found more than 20 different amino acids. And then in terms of the mass of amino acids in milligrams, the amount basically doubled that they were able to detect when they went back and reanalyzed the initial samples. And basically just, there are things that were present at very low frequencies that the equipment in the 1950s just wasn't sensitive enough to detect. You go back in 2008, you could actually catch all that stuff. So that original experiment was actually, if you can think of it this way, more successful than they thought. There's a lot of really cool work going on with these kind of ideas and what conditions gave different molecules and how they could happen on an earlier. A lot of ongoing research on this right now. It's a really cool field. Definitely go down that Google rabbit hole if you are so motivated. But keep in mind what specifically you're responsible for class. Now the other hypothesis, the kind of competing hypothesis with the over and sorry, was that a hand over here? Did I see a hand? Everybody okay? Did I see a hand for a second? No? Okay. The competing hypothesis with over and Haldane is a newer idea. It's called the iron-sulfur hypothesis, sometimes called the iron-sulfur world, because all of the living things would have been in this very specific context initially. So this is the idea that instead of occurring in shallow waters near the surface, these early steps in the process would have occurred deep underwater at hydrothermal vents. Not the ones that are like 95 degrees Celsius, that's too hot, but the warm hydrothermal vents, like these black chimneys that you get way down there at the bottom of the ocean where the water is warm and the temperature is going to provide some of the energy, because temperature that's moving, that's energy. But also it involves iron compounds that are there that can contribute electrons into the system. And as we'll see when we talk about metabolism and photosynthesis, we'll see that electrons can store energy and contribute energy to other molecules. So the idea here is that you've got your warm water, your hot water, things like carbon monoxide, and then like iron and nickel compounds. And they're going to interact in such a way that generates gradients where you have energy cycling and different types of reactions occurring between organic molecules. And there's some really cool findings on this that are much more recent. We're talking just almost all of this is within the last 20 years or so. And so, for example, you can look at hot springs, and you can find very similar environments in hot springs, and you can find precursors to biological molecules, like precursors to amino acids and sugars. You can also look at the environments. It's a little bit trickier to get there, but you can actually directly study this environment way down at the bottom of the ocean, and you can find some really interesting chemical reactions cycling, just like spontaneously. The types of reactions called redox reactions, oxidation reduction reactions, that are very similar to reactions that form the basis for cellular metabolism, reactions that we're going to see in just a couple of weeks when we do cellular respiration. So a lot of work going on both in the field and in the lab for this, and this is an example of just one of the experiments here. So I don't know if you can see what's going on here, but these are basically glass bottles that are upside down, and there's pipes going in through the cap of glass tubes, and they're piping in warm water and like carbon monoxide and iron compounds and those kinds of things, and they're building teeny tiny little pet hydrothermal vents in the lab. And like, I want one of these. If someone made like a hydrothermal vent kit, I would buy one so fast. I want one of these. Look how adorable it is. A little hydrothermal vent on your desk. I want one of these. I'll talk to these guys here from 2015 and see if I can market those. That would be a lot of money. So these experiments are ongoing. There's, I think they're on the right track. I think the hydrothermal vent thing is correct, and I'll share more about what I think when we get to the end today. Any questions about iron sulfur versus Oberon and Haldane?
Could both of them be correct?
Great question. Yeah, could both of them be correct? Absolutely, because we're talking about generating organic molecules, right? Just that first step in the process, you could be generating organic molecules through different pathways in different locations at the same time. Absolutely, yeah.
What makes you
Nice and loud.
What makes you believe that the iron sulfur about this is more?
Great question. What makes me believe that this is more plausible than the other one? Put a pin in that one after we get through all 4 steps. So there's a plot twist I'm going to share with you. We'll get there. It answers your question. Yeah. There's a really fun plot twist after it. It's fun, trust me. Okay, so that's our first step in the process. Abiotic synthesis. Step 1 of 4. We have 4 requirements for this to happen. Low oxygen, plenty of energy, you need the basic chemical building blocks, and you need plenty of time for these reactions to happen. Given those things, you have all the stuff you need. We have 2 hypotheses I want you to know for how this could have happened. Oberon-Haldane and iron sulfur. The big difference being the location and the source of the energy. Could they happen at the same time? Yeah, totally. They could both be correct. Absolutely. So that's step 1 in the process. We have 3 steps left. Step 2 is really easy, so we'll do step 2 next. Is everybody okay? Feeling all right with step 1? All right, let's do step 2. Step 2 is the formation of organic macromolecules. How do we go from just like individual amino acids to polypeptides, for example? This one is probably the best supported, although the next one's pretty easy too. This step is really easy. We have lots of experimental evidence showing exactly how this works. So if you have organic molecules like lipids or amino acids, for example, and you have the right environment, like specific clays that have surfaces that are charged or polar, you can actually have those monomers accumulate on the surface. Now, dissolved in the water, those individual monomers are going to be very low concentration. The likelihood that they're going to interact with each other is quite low. But if you have them accumulating on a surface, like there's a specific type of clay that has negative charges on its surface, so these organic molecules will accumulate on the surface of that clay that's present. It puts a lot of these molecules in a very close proximity with each other, and they're very likely to polymerize under those conditions, especially if we have metal ions like zinc and iron. Getting to the question that we heard a few minutes ago, why do I personally lean towards the hydrothermal vent idea? A lot of metal ions hang out down there. That's part of the answer. It's a lot of metal ions. And they get you through a lot of the hurdles that you need to clear to do these things. So here are some experimental data showing like validation of this step. So what's going on here, this experiment was an hour long, so we're going from 0 to 60 minutes across the x-axis. On the y-axis, this is measuring the turbidity of the solution. Basically, how cloudy is the solution? That's a proxy for how big are the molecules that are present. The bigger the molecules, the cloudier that solution is going to be. So the red line shows the precursor molecules on their own. I forget what is this. This is vesicles. This is going to be for lipids, but you can do this for nucleic acids, you can do it for amino acids, you can do this for a bunch of different things. But in this case, you have just the lipids on their own, and like they'll slowly aggregate with each other, but it takes time. But if you put them in the presence of this volcanic clay, you don't need to know this word, but the name of this particular clay is called montmorillonite. That's a common one we use in these experiments. If you put them together, they shoot up really fast. They just all assemble together on the surface, and then they react with each other and assemble with each other. So that greatly accelerates that process. This has been documented. The data here are from vesicles, but you can also do this with nucleotides, RNA usually, and with polypeptides. So once you have the monomers, as long as you have the environment, which usually involves some kind of ionic, kind of volcanic clay thing, once you have that, the polymerization step is really easy. Yeah? I don't know if technically it's a catalyst. That is a very specific definition. I'm not sure if it's actually acting as a catalyst or if it's just facilitating the reactions by putting things in close proximity to each other. I'm not actually sure. Yeah. I'm sorry? Vesicles. We're going to answer that question in just a second. What are vesicles? But it's just like lipid droplets, basically. Yeah. So that's step 2. That step is really easy. That's pretty straightforward. Yeah.
I just want to say that I agree with what you said about this being more towards the peak. That's the different materials that go into photosynthesis that also feeds the two forms over and say by the many people because they rely mainly on like chemosynthetic bacteria and injections.
Yeah, see, you're getting it. You're seeing where this is going, I think. Yeah, exactly. Yeah. We're gonna get there. Don't worry, we're gonna get there. Yeah, okay. Step number 3. Step number 3 is the formation of protocells. So protocells. Proto being first or early and cell being cell. So a term that we need here is, thank you for the question, a vesicle. A vesicle is a fluid-filled compartment surrounded by a membrane-like structure. And those membranes are, almost without exception, going to be made of lipids of some kind. Do we know of any other fluid-filled compartments surrounded by a membrane-like structure that we encounter in biology class, like all the time? Cells. Yeah, cells. Right? So vesicles, just when they form in the environment, actually are very similar to cells in a lot of ways. So these structures will form spontaneously. You form a lot of different physical conformations. You can get just a bilayer sheet, and then they kind of stack on top of each other. You can get like a sphere with all the hydrophobic tails pointed to the inside. But the one that we care about is this structure where you get a hollow ball. Right? You've got the outside environment, you've got the membrane, you've got the inside environment. Because this is very similar to the structure of a cell, right? That's like a cell membrane structure. We're going to do that in a couple weeks, so we'll get there. That's very similar, right? Here is an actual micrograph of this type of structure forming spontaneously in water. So you have lipids in water, they're going to form these vesicles spontaneously. And the scale bar is really important here. That's UM, it's actually mu, it's a lowercase mu, the Greek letter. And that's micrometers or micrometers. And it's important that we see that it's 10 is that scale bar, because your average prokaryotic cell, your average bacterial cell, is somewhere in the 1 to 10 micrometer. You don't need to know this yet. So in the 1 to 10 micrometer range, a bigger eukaryotic cell, they average in the 10 to 100 range. So these vesicles that form spontaneously, you can see, they are basically the size of cells. That's cool. Then we have cell-like structures that are approximately the correct size, forming spontaneously once the lipids are present in the aqueous environment. That's a really cool finding. So why do we call these things, these composed structures, protocells? Well, protocells are aggregates, so you're combining a bunch of things together, they're aggregates of abiotically produced organic macromolecules. And that abiotically produced is really important. When our bodies make cells, those are not protocells. Like our red blood cells, for example, don't have a nucleus. If you don't know what that is yet, don't worry. But like if you do, your red blood cells are missing some parts. But we don't consider them protocells because they come from other cells. Protocells are by definition abiotically produced. Now they share a bunch of traits with living cells. And we're going to go through this list and we're going to see that this applies to cellular life. So like having an electrical potential across the surface of their membrane. In other words, there's going to be more ions on one side of that membrane compared to the other. That's going to be really important next semester when we talk about the nervous system and muscles, right? Having those ions in different concentrations, that's a big deal. We also see that protocells, like real cells, will absorb materials including water. So they do this thing called osmotic swelling, which is a really fancy way of saying they absorb water, and when they do that, they get bigger. We'll talk about membrane transport, and we'll see what osmosis is and how it works and why. For now, just know the term. Osmotic swelling. They absorb water, they get bigger. Because that membrane surrounds this structure, this protocell, some things can get through and some things can't. So the internal environment is chemically distinct from the external environment, just like living cells. And this is the really fun part, protocells will divide in two if they get too big. Now this is not cell division. It's a purely physical process where the lipid bubble basically gets so big it becomes unstable. And when that happens, it just kind of blobs into two smaller bubbles that are approximately 50-50 in terms of the contents of the previous one. This is interesting for a bunch of reasons. One, it's not cell division, but it sure looks like cell division. The other thing is you'd think it would be a really messy process and like all the stuff would leak out when it splits. Turns out that's not the case. It's actually a really efficient process that keeps most of that internal environment the same, going from one big protocell to two small ones. Okay, so protocells don't have all the traits of a true cell, but they have a bunch of the traits, and that's important. We okay with protocells? Questions? That's step 3, forming protocells. Yeah?
When you say
Organic, so like carbons, so lipids, carbs, proteins, stuff like that. Stuff we just talked about previously. Stuff that is carbon, yeah. Yep. Okay. Important thing. Lead into our next requirement here, our next step. What are we missing? Heredity. There's no mechanism of heredity in protocells. There's no DNA, no RNA, no way of saying, I am this thing, let's specifically make copies of that thing, right? There's no mechanism of heredity yet, which is the lead-in to step number 4. The step number 4 is going to be, anybody remember what step number 4 was? Just shout it out, you got it, just shout it. Self-replication. That's where the heredity comes into play. Self-replicate, you copy of yourself. So now, this is what's going on in living cells. Like right now, this is how living cells work. Right? You've got your genetic information stored in DNA, and then that DNA gets transcribed. Don't worry about these specific words yet. This is kind of background. We're going to do all this later. The DNA gets transcribed into RNA, and then that RNA gets translated into a protein. We're going to do that in like October. We're going to talk about this arrow and that arrow a whole lot. All right, but the key thing for today is RNA, right in the middle of this process. We think that RNA was the first replicating molecule initially. It would have been the first nucleic acid to appear in living systems, and it would have been the first replicating molecule, or sometimes people just say the first replicator within living systems. It would have been an RNA molecule. And we have some very good reasons for thinking that is the case. Has anyone ever heard the term RNA world or RNA world hypothesis? Has anyone heard that term before? No? Interesting. Great. We're having so much fun. So, this is going to be the focus of the appearance of self-replication. Because RNA, in terms of what it's capable of doing, is very different from DNA, it's very different from protein. Okay? And the idea that RNA would have started this all off is called the RNA world hypothesis. Now I want to just clarify something. Most people today who are kind of on board with what we've been talking about so far, kind of more or less agree with these steps as the most likely pathway to living things, they don't subscribe to a strict RNA world where it was just RNA and no polypeptides or proteins involved. Most people are going to say there would have been some combination of RNA and protein interacting with each other. And I think that's probably correct. I think, that's probably correct. The thing that we want to focus on with the RNA in the RNA world hypothesis is not the idea that it would have been just RNA and nothing else playing a role here, but it's the idea specifically that it's the first replicating systems used RNA to do two things. One, store the genetic information, and also to copy that genetic information. Right, so RNA is capable of doing both of those things. DNA and proteins can't. DNA and proteins would have been incorporated later. DNA strictly for storage, proteins strictly involved in replication, but RNA is a unique molecule and that it could be involved in both. So we think that that had to be the starting point, probably with some help from some short polypeptides is what we think. But we're going to focus on the RNA. That's what matters. Are you okay with that idea? Yeah. Go ahead. The louder? Yeah, so we're going to talk about CRISPR when we do DNA technology later in the semester. This idea is much older than the discovery of CRISPR. CRISPR is a relatively new thing, last, I don't know, 15 years, maybe? Was it right? 15 to 20 years, something like that? Yeah, 20-ish years? Yeah, this idea is much older. This is a much older idea. No, this idea was actually, we'll talk about it in terms of the popularity of this idea. But it's a different trajectory from CRISPR. It's a totally different thing. They both involve RNA, but this is a different kind of thing going on. Okay. Now, the key word, the reason RNA is the linchpin of all of this, are these molecules called ribozymes. Ribozymes. A ribozyme is an RNA molecule with enzymatic properties. In other words, it's a piece of RNA that can speed up a reaction like proteins can. And it's RNA that can do the job of an enzyme, but in all of our cells right now, those jobs are done by proteins instead. So for example, you can have ribozymes, you can evolve these in the lab to cleave an RNA molecule. You can take an RNA molecule and you can make a ribozyme that cuts that RNA molecule into two at a specific place in the sequence. You can also make a ribozyme that catalyzes RNA polymerization. It will be a little single-stranded bit, and it will catalyze the synthesis of a complementary strand, of a double-stranded molecule. Right? So you can have an RNA enzyme that does various biochemical reactions. You can't have DNA enzymes. That's not a thing. I shouldn't say you can't, because it's biology and you never know. But we have yet to discover any way that DNA can function as enzymes. We know that RNA can function as enzymes. And that's the key idea here. Okay, so here's an example of this. You don't need to write this down just to show you. This is an older paper. This is from 2002. And this is an example of a lab-created ribozyme. They evolved RNA molecules in the lab through a really cool process called in vitro evolution. You don't need to know what it is or how it works, but they basically evolved these little RNA sequences, and each of these lines represents a single-stranded RNA molecule, and they can catalyze the reactions of going back and forth between all of these states. Right, so it's a lab-grown replicating ribozyme. This is from 2002. There's been a bunch of examples of this that have been developed in the lab. Key thing is ribozymes are RNA molecules that can catalyze reactions. So we can put these two steps together to get something that sure looks like replicating, dividing cells. Okay, so we're going to put our protocells and our self-replicating RNA together. I'm going to show you little pictures here that are going to pop up. I'll guide you through exactly what's going on in the pictures, don't worry. So we're gonna have RNA polymers are gonna be shown in red, and the vesicles made of lipids are gonna be shown in blue. Okay, now the key thing here is that when you have bigger molecules inside your vesicle, water is gonna rush in, and remember what we said a few minutes ago about protocells, when water goes in, they'll grow, they'll get bigger until they get unstable and they split. Okay, so we're going to start this right here with two competing protocells. This one right here, this little squiggle of RNA, that represents a ribozyme that can make copies of itself. This one down here, it's got RNA in it, but it's not doing anything. It's not catalyzing the reactions, it's not copying itself. Okay, so this cell has the self-replicating ribozyme, this one doesn't. Everyone see that? I called it a cell, it's a protocell. Everyone okay with what we're starting with here? Okay. So what's going to happen is that as this ribozyme replicates, what it does is it takes the other RNA that's present in that protocell, and it's going to rewrite it as copies of itself. It's going to catalyze the formation of copies of itself. That's self-replication, that's what that is, right? It's replicating itself. Now this protocell right here is going to absorb water because it has these bigger molecules in it. This one down here will not do anything, it's just going to stay the same. Okay, but this one's going to get bigger. Okay, so that one over time, it will absorb water. As it absorbs water, it's also going to absorb lipids from the surrounding environment. Right, you're going to have just lipids hang out. Those lipids are going to be incorporated into the growing protocell. Now, where else is this protocell going to get lipids? It's going to steal them from that one. So if you're not growing, you're going to have your lunch money stolen by the protocells that are growing. Okay, so the ones that don't have the replicating ribozymes are going to shrink over time. They're going to lose both their lipids, and when those lipids get absorbed, the RNA in there is going to end up in here. And that RNA is what? That's raw materials to make more copies of those ribozymes. Let's get more of the raw materials to make more copies. Now what happens when this protocell gets too big? It splits, it gets unstable. So now it's going to split into two protocells, and like I said before, when it splits, you have to keep that content the same. So some of those ribozymes are going to end up in each of the daughter protocells. So now look at that. That sure looks like a cell that's doing cell division. It's not. It's not a complete cell, it's missing stuff. What's the big thing that's missing that we haven't talked about at all right here? Well, the RNA fulfills the genetics, but I like what you're thinking. Proteins, what processes? What are we not making any of in here? We just got lipids and we're doing some RNA. Energy, there's no metabolism, right? So this is all just purely physical, chemical processes. Yeah, you had it, pretty shy. This is all just physical, chemical processes using energy from the environment, right? It's not a cell, it's not doing metabolism and generating its own chemical energy to accomplish its reactions. But it has a lot of the components of cells and cell division, and this is really important. It will be a little while before we get there, but like this is how natural selection works. This is competition for resources and selecting for the things that replicate the fastest. That's what's going on here. This is evolution. So once you hit this threshold of activity of the environment, evolutionary processes, evolutionary mechanisms take over. That's what's going on. Questions? How do we feel? Okay. Now this has some really interesting experimental support. We've actually documented a bunch of steps of this, including the growing and the dividing. And here's a really interesting one. If you do some of those polymerization experiments I showed you way back in step 2, if you do those experiments with different materials together, like you do it with lipids and with RNA at the same time, you end up with a vesicle. You can just barely see the vesicle boundary. It's that vague circle in there. Here, there it is. You have that vesicle boundary right there. And then inside that vesicle, we have RNA polymers inside the vesicle. So if you have the conditions to induce polymerization that we saw in step 2 of the process, plus lipids and RNA monomers, you end up with RNA polymers inside the lipid vesicles. You end up with protocells with RNA on the inside. If that RNA can act as a ribozyme that can facilitate making copies of itself, there we go. And what we know about those ribozymes is they can be really short. They can be just 100 or 200 nucleotides long, and they have quite a bit of activity. So that's a really, really interesting demonstration of some of the steps in this process. Now, you're probably wondering, what about DNA? I will tell you, this slide, you don't have to know this. This is just to answer the question that one of you is thinking, at least one of you is thinking, okay, what about DNA? This is the answer to that question. You don't have to know this. This is not something we're going to ask you about. So, RNA came first, and it would have been single-stranded, just one chain of RNA. Now, at some point, it becomes double-stranded, probably due to doing this thing called a hairpin. You don't even know what it is, doesn't matter. And it becomes double-stranded, and that's good because it's more stable. If it's more stable, it's going to last longer. So ultimately, you're going to have a lot of double-stranded stuff just because it lasts longer. Then, we think DNA gets incorporated, but at first it's just one half. So you have like an RNA-DNA hybrid double helix. And once you have that, now that RNA bit has to let go of the DNA to do its enzymatic stuff. But the DNA is more stable for storage. You can see how ultimately those two functions get partitioned out into separate molecules. So that's the idea. That would have been RNA first, then DNA would have come later. There's a really good paper on this. Ask me about it if you want. It's one of my favorite papers on this RNA to DNA transition. Beyond the scope of this class, it's really cool. Now, another question that always comes up is, why isn't life constantly forming? We still have hydrothermal vents, right? We still have hot springs, so why isn't life constantly forming? The answer is because cells already exist in all of these places. You can go to deep sea hydrothermal vents, you're going to find microbes living there. You can go to hot springs in Yellowstone National Park, you can find a lot of microbes living there. Any organic molecules that are going to form spontaneously in those environments, they're not going to accumulate for 100 million years because there's already living things there that are going to gobble up anything useful that might spontaneously appear in the environment. So this process that we just talked about, this is a one-shot deal. Once there's cellular life present in these environments, wherever it happens, those cells are going to take up anything that have previously accumulated and contributed to cellular life. It can't accumulate anymore. That makes sense to everybody? Again, it's not something we're going to ask you about, but people always ask, is this still happening? No, and it's because life is already there. So here is the summary of abiogenesis. Our 4 steps again. Abiotic synthesis of monomers, and we talked about the requirements, the competing hypotheses there. Then we have the formation of organic macromolecules. We have the formation of protocells, and finally, self-replication. Once you put self-replicating molecules inside protocells, you're really, really close to, what we think of as cellular life. Now, what are we missing, again, when we get to that step, once we put all that stuff together? We're missing metabolism, right? So we're not there yet, but we're pretty close. Your question?
Yeah. So if somebody were to take the proper environment
Yeah.
into space, then wouldn't that give a
Put the proper, so to deep sea hydrothermal vents and put in space and left them there for 100 million years. Yeah, the problem becomes the time frames, right? It becomes really hard because of the time frame. So what we do is we can purify these things and simulate it in the lab, right? And because it's purified, we're going to hope we'll be able to accelerate it because you don't have all the sine reactions and backward steps and everything. That's the idea of how a lot of this research works. Now, we're not going to get to the history of life on Earth today, that's fine. So we're going to start with part 2 next time. It's not a long section, but I don't want to rush through it because I want to tell you, I want to give you the plot twist for all of the stuff that we just talked about. So here's the plot twist. There's still 6 minutes. Don't start packing up yet. Here's the thing. This is why I love science. Everything I just told you, not everything, but this overall process that I just told you that you need to know that we're going to assess you on, I think this is wrong. I don't think this is how it happened. There's a newer idea called metabolism first. You don't need to know anything about metabolism first. It's a new idea, newer. It's definitely a minority position among biologists, probably 20, maybe 30% for the metabolism first camp. But 10 years ago, that was maybe 5%. 20 years ago, it was maybe 1%. Metabolism first is winning over a lot of people because the way metabolism first works is, and this gets back to your question, it takes place in deep sea hydrothermal vents. And what happens in deep sea hydrothermal vents are chemical reactions. They happen spontaneously that are very similar to some of the central reactions in cellular metabolism that exist right now. And so what the metabolism first people think is you have the chemical reactions happening first, and then different subsets of those reactions became compartmentalized in lipid vesicles. So the cell-like structures came later, the ribozymes and the heredity came later. It was actually the metabolic reactions were first, and they were environmental reactions, and then they became kind of encapsulated within cells later on. That's the metabolism first hypothesis, and I think that's where the best data points right now. That's a newer idea. You won't find that idea in textbooks. It'll be 10 or 15 years before that idea is in textbooks. But I think that's where the research is going. If you want to ask me about that, ask me after class, because that's, again, you need to know that. Get me after class, because I want to just get to the practice question for today. So we're going to skip part 2. We're going to come back to this next time. Again, it's very short. It's only like 6 slides. We're going to come here. Oh, hang on. We're going to come here. Here is your practice question for today. What we covered. The outcome is sequence abiogenesis. Correctly sequence the following steps in the process of abiogenesis.
Basically, you only
Talk it over with each other. Figure it out.
whatever I'm clear, like you don't need to do this.